Good evening, everyone. It's a pleasure to welcome everyone here tonight to this special 10th anniversary of the With Insight program, A Foot in Both Worlds. I'm going to put up my first slide. Hopefully this will go okay. Okay, hopefully everyone can see this. Tonight, we're going to be talking about the past and present of the Koch Institute Clinical Investigators program, which focuses on the intersection of laboratory research and clinical practice. I'm Michael Yaffe, director of the Koch Institute Clinical Investigators program, and like the investigators that you will hear about tonight, and like our Koch Institute director, Matthew Vanderheiden, I am also a physician scientist who currently both conducts laboratory research and conducts and sees patients directly. Now, the Clinical Investigators program is part of the Koch Institute's clinical partnering strategy, which is an important outgrowth of the Koch Institute's mission. The Koch Institute, as I suspect many of you are aware, is one of only 7 basic cancer centers designated by the National Cancer Institute. And we've been an NCI designated cancer center since 1974. Interestingly, the Koch Institute and its predecessor, the CCR, or the MIT Center for Cancer Research, is an NCI center without a hospital. And so you might ask, what in the world are we doing with a clinical investigators program? Some of you may recognize some of the people on this slide. You can see in the bottom left, that's Phil Sharp. In the middle of the bottom, you see Bob Weinberg on the right and Salvador Loria, the founder of the Cancer Center. And then this is the old building in the upper right that the Cancer Center used to occupy, which I've been told is actually part of the old Necco candy factory. Now, this is the 50th anniversary of the National Cancer Act. This was an act that was signed by Congress in 1971 under President Richard Nixon that declared a national focus on cancer. It provided legislation that was able to bring about the infrastructure that was needed for the country to focus on this disease. And this was a bit contentious at the time, and not everyone agreed that it was a priority. And some people thought we were spending too much money on a disease, and some people thought that we weren't spending enough money on a disease. More importantly, you have to remember that in 1971, we didn't really understand cancer biology, let alone how to target it. And it's only now that there's really a role, I would argue, for a clinical investigator type of position in a basic cancer center like ours. And that's part of the natural evolution that we've seen in the last decade or so in MIT's approach to cancer research, bringing in basic scientists, engineers, and systems biology-based thinking. So the Clinical Investigators program that you'll hear about tonight emerged as the Center for Cancer Research transitioned into the Koch Institute, and the need arose in the broader field to better understand the real clinical problems. Now, the Koch Institute is not the Broad. We sit at a particular nexus in the cancer research field because we're not the Broad and we're not the Dana-Farber. The Broad, as I suspect you know, is largely focused on genetics. And the Dana-Farber, which is largely organized around treating patients based on where their tumor originated. But here at MIT, as I've mentioned, we sit in a somewhat different place, both physically and in terms of our outlook. Because we're MIT, we have an engineering and systems approach to thinking about tumors. Because we used to be the CCR, we have a deep biological knowledge about the tissue of origin of tumor cells, about the genetic mutations that underlie them, and about the lineage in which the tumor cells evolve, which controls the biology and the behavior of the tumor itself. And finally, of course, because we're MIT, we have the ability to think about this not only qualitatively, but in a very deep, quantitative and systems-based way. We now know that with cancer, it's not just genetics and it's not just lineage and it's not just location, but it's all of those things together and how we can act on them. Now, one of the great dangers, I think, that we face in doing the type of science that we do at MIT versus doing clinical research is falling into the trap of only doing what it is that we think is particularly interesting. On the other hand, if we focus strictly on clinical problems, then that have immediate clinical application, then we miss out on important foundational knowledge, which will ultimately inform future clinical applications. But at the KI, we don't see this as a one or the other proposition. In fact, we see this as an and function, where we can do both foundational science and translational work. But in order to do that, we have to really understand what the clinical challenges are so that we can focus our research towards the clinical solutions. And in order to accomplish that, the KI Clinical Investigators Program is part of a constellation of programs, initiatives, and collaborations at the Koch Institute that are aimed at keeping our work oriented to what patients and physicians need. Whether this is presenting clinical case studies of specific tumor types or working directly with our clinical colleagues, either through the Bridge project that you see indicated here, which allows MIT to bridge with a consortium of Harvard teaching hospitals, or our ongoing collaboration with the Mayo Clinic, or our ability, largely through the Clinical Investigators Program, to bring in practicing physicians who can tell us about the vexing clinical challenges that they face. These are the types of opportunities that we use in which we try to engage clinicians in order to understand better the clinical perspectives on the diseases that we study. Now, you're probably thinking, why not just team up with established clinicians? And part of that is because part of MIT's mission is to train the next generation. We are committed to the professional development of future physician scientists who are focused on the treatment and understanding of cancer. And so rather than telling you about all these people, I think the best thing to do is to let them tell you themselves. So in a moment, I'm going to allow them to introduce themselves, but I want to give you a quick overview of tonight's program. We're going to hear briefly from each of our speakers. We've highlighted 4 of the 5 clinical investigators here. Scott Floyd, a radiation oncologist, Dr. Ho-Jun Lee, a pediatric oncologist, Dr. Salil Garg, a pathologist, and Dr. Alice Shaw, a medical oncologist, all of whom will tell us about the aspects of the KI's clinical investigator program and whether it helped or did not help them. After we allow them to speak, then we'll have some discussion time, and then we'll open the floor to your questions, which you can enter into the Q&A window at any time. And so with that, I'm going to stop sharing my screen, and I'm going to invite the first of our clinical investigators to introduce themselves and tell you their perspective.
Thank you so much, Mike, for that great introduction to the program. I'm very excited that the development office organized this event tonight. It's a great opportunity for all of us to share what we've been up to. So my name is Ho-Jun Lee, and as Mike said, I'm a pediatric oncologist. I practice at both Boston Children's Hospital and the Dana-Farber Cancer Institute. And within pediatric oncology, I subspecialize in bone marrow transplantation, specifically for high-risk cancers, especially high-risk leukemias. And so really my research focus all has to do with those very difficult to treat leukemias, which are also by extension a model type of refractory cancer that we hopefully can also develop novel paradigms or ways of thinking that can also be applicable to other difficult to treat cancers. And so what I work on at the Koch Institute, or what my lab is focused on, there were 2 main goals that I had when I arrived. One was to understand how these incredibly refractory and difficult to treat cancers hijack some of the existing blood stem cell programs that make them very quiescent and very inactive until bursting into action to produce more cells, and how those programs can make them resistant to the chemotherapy regimens that we have currently. And additionally, another goal I had was to study how those cancer cells in the blood, how they ignore the normal signals to differentiate and become the red cells, the platelets, and the white cells that are normally circulating around. And being at the Koch Institute has really opened my eyes additionally in terms of my research group focusing on additional research questions. And this idea that Mike presented about how we're at the nexus between biology and engineering has all of a sudden opened up many new avenues for us to study targeted drug delivery or approaching the cancer progression process in a very sort of systems biology, quantitative manner. And I don't think any of that would have been possible had I not become immersed in the last 18 months in the environment of the Koch Institute. And so certainly my clinical practice informs the types of cancers that I study in the research realm, but additionally, the types of collaborations that I've been able to generate through being a principal investigator here, I think I probably no less than a third and probably half of the faculty within the Koch Institute I've had collaborative interactions with, whether it's on the same floor, Angela Kohler or Michael Birnbaum, or on different floors, Sangeeta Bhatia, and we're starting up some new collaborative ideas with Scott Manalis. And really the list goes on, and none of that really would have been possible if I had solely been performing research over at the medical center in the Longwood area in Boston. And I like to think that even though my research program and group have benefited greatly from the environment, as Mike alluded to, I hope that the presence of clinicians such as myself has benefited others. And certainly the joint group meeting that I run with Michael Heeman, he does a lot of work, or his research group does a lot of work on blood cancers and screening for blood cancers using novel genomic strategies. And certainly my clinical practice hopefully has been benefit there, but also in terms of the seminar series that Salil and I currently run, where every month we bring in a clinician at the very cutting edge of clinical practice to let the labs here know these are the important problems that are preventing us from treating patients more effectively, and hopefully giving a lot of new ideas for the other labs here to study. So that's sort of, I think that was my directive to talk about how we hopefully bring a collaborative and clinical feel with us with our research programs. And I guess I will hand off to Salil, who will talk about his experiences and additionally how about the great amount of mentorship that we receive as junior PIs through this program. So Salil, I will hand it off to you.
Thank you, Hojin. That's really fantastic. So I'll start by introducing myself just a little bit. So my name is Salil Garg. I'm like most of the clinical fellows an MD PhD, and I practice as a molecular pathologist over at Mass General Hospital across the Charles River, and then run my research lab over here. And so I was told to talk just a little bit about what my lab does and what we're interested in. And so, as I said, I practice as a pathologist. And so, pathology is really a support specialty. And, our job is to render diagnosis of cancers. And I noticed as a pathologist, and I think we're all familiar with this concept, a lot of times patients get treated for cancer, but the cancer comes back. Treatment fails to eradicate all of cancer. And what we do in molecular pathology is we look at the mutations in cancer and we try and say, okay, what genes are mutated? Can this help predict treatment or help tell our oncology colleagues, how to target therapy at a particular cancer? And I've been really embarrassed as a molecular pathologist that oftentimes we don't really have good mutation information for a lot of tumors. It doesn't seem like there are a lot of changes sometimes, or at least when the cancer fails treatment, there are no new mutations compared to when a patient first came in with cancer. And being a pathologist without a diagnosis is kind of like being a radiologist without a picture or a cardiologist without a stethoscope. You're kind of like, well, what am I doing here? What's my role in this team if I'm the pathologist and I can't render a diagnosis? And so we've tried to understand what's going on in these tumors that don't necessarily have new mutations. And what we think is going on is that, they switch kind of their program of how they're behaving. And basically, we think a lot of those in more scientific terms, we might say they're sort of state switches or epigenetic switches. But basically, our underlying hypothesis is that the reason that tumors are doing this is because these are natural programs from development, from when we were all embryos and trying to make lots of different types of tissue and lots of different types of cells in the body. And so we're trying to sort of systematically map cancers to different programs or different switches that take place during normal embryological development. And of course, when we develop as embryos, hopefully we're not accumulating tons and tons of mutations. And so we think those processes are kind of analogous. And that's one of the main thrusts of my lab. And the other piece that we're building on is to then take how we're understanding some of those switches that are happening and actually try and build machine learning or artificial intelligence-based diagnostics that can really help in the clinic as well. And of course, it seems like we're at MIT. What better place to do artificial intelligence and machine learning? We're just getting started on that, but I think it's an exciting area. And so for me in my lab, the clinical investigator program has really been fantastic. I mean, I think what we work on really informs this kind of circular process of discovery, from what do we see as problems in the clinic where we're not able to render as specific diagnoses as we like to help inform our oncology colleagues, and what we see as problems on the basic science side. How can we understand the connections between cancer and normal developmental biology, and what might be going on in cancers as normal developmental programs break, and are those things even related at all? And I guess Hojin mentioned I was going to talk a little bit about mentorship. I mean, I think one of the great things about being in the immersive environment of the Koch Institute is we have, great mentors like Mike Yaffe, great role models, like Scott Floyd and Alice Shaw, who are going to talk after me, who have come through this program before us. And the new director of the Institute, Matt Vanderheiden. It's fantastic to see folks who have gone down the physician scientist track before us, and laid the groundwork, and then to try and pass that on to the trainees in our own labs. I think I can speak for all of the clinical fellows in the program. Our labs tend to attract people on the MIT side who are thinking about medicine and medical problems a little bit, and and many of them interested in going into medical school or at various stages of training. And it's great to be able to pass on, the little tidbits of wisdom we get along the way in that fashion. And the Institute's been really great for that. And I'm hopeful as we emerge from, and the world emerges from the COVID shutdowns, that we'll start to get more of that kind of informal interactions back as we can be together in person more. But really, the program's been great for me, and I'll be happy in the panel to chat with everyone and take any questions you all might have. So with that, I'll pass it off to Dr. Floyd.
Hi, good evening, everyone, and thank you so much for taking time to join us for this program to tell you about what I think is a very, very important part, well, personally for me in my own life, of training, what helped me develop to be a physician scientist, really, the clinical investigator program at MIT. Important to me, but I think important to MIT in general in the larger field of cancer research. The idea behind the clinical investigator program and the environment of MIT really, I think, has the potential and has already, as I hope I can show you, made significant, important impact in patients' lives, in the lives of doctors treating cancer. There are discoveries happening right now. There are discoveries that happened when I was a clinical investigator that make real concrete impact on the lives of patients. And that's very important, and something that I hope, and I think we all hope, can be perpetuated and continued and expanded. So what I thought to do is maybe give you just an arc of the journey that I took to get where I am right now. And as you see that arc, I think the clinical investigator program will be an obvious, very, very important part of how I ended up where I am now. And through that, there's two overarching themes that I think will come through that you've already heard a little bit about. from Hojin and from Sunil. First, the idea of environment, and environment, most importantly, perhaps the people that are around you in that environment that shape you into who you are. And then the other idea is collaboration, because I think that that MIT, the Koch Institute, and the Clinical Investigator Program in particular encapsulate the very best of environment, people, and collaboration. So now I'm at Duke University down here in the sunny South, where I'm a radiation oncologist, and my clinical focus is in brain tumors. And that's a focus that I've had for a long time, since I was in medical school, where I was interested in the brain always, but brain tumors, I came to learn, are a particular problem because so many of them are so hard to treat. And so the main focus of what I'd like to be able to do someday is say that we've really made progress in brain tumors, and that we've been able to add to the treatments and to the lives of the patients that suffer from brain tumors, because it's a major problem for those patients that have brain tumors. So I remember the first time, so I'm doubly lucky because I was a clinical investigator, but before I was that, I actually worked in Mike Yaffe's lab for a number of years. So I remember the first time I met Professor, Colonel, retired Michael Yaffe. It was at a symposium there at the Public Health School at Harvard. They have a symposium every year called the John Little Symposium, and it's about DNA damage. And as a radiation oncologist, that's what we're doing in the clinic, causing DNA damage. And that formed kind of a nidus of the things that I've been interested in for studying in the lab for a long time. So I went to this seminar, and at this seminar, Professor Yaffe gave this phenomenal talk about the work he had just done, describing the function of a very important protein, the BRCT domain, parts of proteins that are involved in BRCA mutations that many of you probably have heard about, that cause so many cancers, breast cancers, ovarian cancers, and other cancers. So describing how, he had discovered how these things work, it just lit me on fire. And so I remember running down after his talk and saying, I need to work in your lab. And he graciously said, okay, crazy man. And he let me do it. And so I was there for a number of years. And at the end of that, we had developed a story that was very close to being finished, but wasn't quite finished. And he said, you should look at this opportunity that we have at the Koch Institute that I'm starting to head. We've had one person come through. It was Alice Shaw. You should think about doing this as a clinical investigator. And I said, okay. Well, then I went off and I talked to Alice Shaw, and Alice was at Mass General just across the way, just across the river there. And she had worked in the laboratory of Tyler Jacks for a number of years, and had, moved into clinical research. And she talked to me about the program and how it had helped her. And I thought, okay, let's do it. And we got involved in the program, and it was absolutely critical because it allowed me the time to finish that project with Mike, where we made an important discovery about how radiation and DNA damage in general can be shaped by molecules called bromodomain proteins, which there are now inhibitors for that are coming to the clinic. And so that experience gave me the time to finish that story, get funding, and then move to Duke, where I was recruited by people that I had actually met again at the Koch Institute, David Kirsch, who had also worked in Tyler Jacks's lab. And so you see the way people can influence you so much. And David talked to me and said, why don't you come to Duke and carry on what you're doing? And so that's what I've done. Built the lab here that now focuses a lot on brain tumors. And what we're doing in the lab now is trying to develop systems that allow us to keep individual patients' tumors alive in the lab so that we can actually find new therapies and perhaps even personalized therapies that work for these patients in the lab. And all of this is made possible through the people I met, the collaborations I had, and the environment that is the Koch Institute and the opportunity of the clinical investigator. And I want to hand it over to Alice Shaw, but before I do, I want to just show one quick slide, and that is, I don't know if I can do it. Yeah, here it is. Because I see patients, in the clinic, and every time I see one of these patients, I think of Alice because they have a specific mutation that Alice was essential in moving forward the therapies that we now use. This is a patient with brain metastases. We've been seeing this patient for a number of years. And, when we first saw her, it was very worrisome. You can see all these spots in the brain. There were a number of them. This is just one slice, and many slices, there are many tumors. And using the drugs that Alice was essential and moving into clinical practice, this is what her brain looks like. Wait, let me get the thing to work. Ah, there we go. You can hopefully see it. There, that's bigger. What it looked when we first met her and what it looks like today. All the spots completely gone, and we never had to give her radiation, which is amazing. So with that, I want to hand it over to Allison. She can tell you a little bit about her journey and some of these things that she's been quintessential in giving us the tools that we need to really help patients.
Hi, everyone, and thank you, Scott, for that very nice call out. So I'm Alice Shaw, and it's great to be here tonight and to see so many of you interested in learning about our this KI Clinical Investigator program. Just by way of introduction, I was in the MD PhD program at Harvard Medical School and finished up a long time ago, 1999, and then went on to internal medicine residency at Mass General and clinical fellowship in hematology oncology at Dana-Farber and Mass General. And as you heard from Scott, for my postdoc, I was here at the Koch in the JAX lab and spent over 5 years studying mouse models of lung cancer. And after my postdoc, I then transitioned to a junior faculty position at Mass General in thoracic oncology, which focuses on any cancers arising in the chest. And at the same time, I also became a clinical investigator here at the Koch. Now, this was back in about 2008 to 2010, and that was really just the start of my career, and actually coincided with, when this field of lung cancer was just starting to explode, based on a number of really seminal discoveries, including the discovery of oncogenes, cancer-causing genes, as the drivers of cancer and specific subsets of lung cancer patients. And what was remarkable is that we were just starting to have drugs enter the clinic, and these are targeted therapy pills that could turn off these oncogenic drivers and induce dramatic and really rapid responses in patients like the one Scott showed. And we never had medicines like this before. I mean, chemotherapies just don't induce responses like that. And so that was really the start of precision cancer medicine, which has transformed not only lung cancer, but really all of oncology today. Now, even though we saw these amazing responses, and we actually often call them Lazarus responses because these pills can really bring patients back from the brink of death. But despite these amazing responses, we were also starting to see that cancers can come back after a period of time. And for some patients, the cancers come back after just a few months, but for other patients, the cancers come back maybe after a number of years. And so as a clinical investigator at Mass General and at the Koch, I wanted to study why cancers respond to targeted therapy pills, at least initially, and then why and how they change in order to become resistant. And so to answer these questions, we started by studying traditional cell lines in the lab and growing them in the presence of the targeted therapy pill. And usually for many, many months, we would actually start to see resistant cancer cells emerge. And then we would study these resistant cells in depth to try to discover what was driving resistance. And, this is a very informative approach, actually. We were able to identify clinically validated mechanisms of resistance, but it was very slow and painstaking. And it became clear that we couldn't fully capture the breadth of resistance seen in the clinic. And so at this time, we decided to really understand why patients become resistant, we had to study resistant cancers from the patients themselves and apply that same scientific rigor and line of questioning to clinical specimens, just as we did to lab-based projects. And so this led us eventually to developing a repeat biopsy program at the hospital that allowed us to sample resistant cancers from patients and then to thoroughly interrogate them at the genetic level to identify genetic causes of resistance. And with these tiny, tiny little samples from biopsies, we also learned how to grow them in petri dishes in the lab in order to generate many new models of resistant cancer, which have been super helpful for us in terms of defining mechanisms of resistance and actually now allowing us to test new therapies and combinations that are designed to overcome resistance. And so based on all of this understanding of response and resistance, and this is work over a decade, but our work in the lab really did contribute, I think, directly to the development of many new agents, which have gone on to receive FDA approval, including the first generation targeted therapies, Alkori, as well as now second and third generation drugs, with each new generation, really building on our understanding of resistance and leading to better outcomes for patients. Now, at the same time, our observations in the clinic also help to highlight pressing needs for patients, and that actually was really helpful in informing how drugs need to be designed. And so actually, Scott, the example you showed is really relevant to what I wanted to highlight, because I think that, as you said, unfortunately, these patients can develop brain metastases. And they can be very, very difficult to treat with traditional therapies. And knowing this and seeing this in the clinic as a frequent occurrence, the latest generation of targeted therapies were designed to be highly brain penetrant. And now we're seeing phenomenal clinical outcomes, responses like the one Scott showed with these newest targeted therapies. And these can not only address resistance that develops in the body, but also treat and prevent even metastases in the brain as well. So I worked in this field of targeted therapies and drug resistance for over 12 years. And during that time, I became director of thoracic oncology at Mass General and a professor of medicine. But, more recently, I decided that I wanted to do more and to apply what I had already learned to other areas, and actually just to really learn how to develop drugs for cancer patients. And so at the very end of 2019, so about 18 months ago, I moved from Mass General to Novartis. And specifically to NIBR, which is Novartis Institutes for Biomedical Research, which is just down the street from the Koch. And at NIBR, I'm leading early clinical development in oncology. And actually right now, I'm also the interim co-head of NIBR Oncology. And at NIBR, we're developing multiple different therapeutic modalities across all different cancer types. And it's been really, really, really exciting. It has been about 18 months since I joined, and I've learned an incredible amount and have been actually really impressed by how directly relevant all of my research experiences have been to this role, especially the translational research. We often talk about translating discoveries to the clinic in academia, but actually in industry, that's exactly what we're doing in the form of new medicines. One other thing I wanted to mention is that in addition to my role at Novartis, I'm still actually maintaining my clinic at MGH. I have a half day per week at the hospital because I really do believe that clinical experience and perspective is essential and actually grounding for those of us who are developing new medicines. So that's a very quick recap of my career, and hopefully you can tell that my first experience here at the Koch was really foundational and helped set me on the right path in terms of integrating science and medicine to have the greatest impact on patient care. So with that, I'm going to turn it back to you, Mike.
Thank you very much, Alice. I want to start by thanking Drs. Ho-Jun Lee and Salil Garg and Scott Floyd and Alice Shaw for taking the time out of their day to share with us what their experiences were like, and especially to highlight, as they have so nicely done, the evolution that happens when people start the program after a short or longer postdoctoral period and then transition to their own independent careers. And so I would encourage anyone in the audience that's interested to please use the Q&A box to ask any questions. But I've got a couple questions that I've already thought up that I'd like to open up for roundtable discussion. In particular, what I'm going to ask is all of our panelists now to turn back their cameras back on. And what I'd be interested in understanding is, how has the clinical investigator experience been different for you than at other stages in your training as a physician scientist? How is this different than what you had seen or done as a medical student or as a resident or as a PhD student or even as a postdoc? So I will open it up.
So I think I'm probably the most recent close to training, since I'm the newest of the clinical investigator program, and without going too overboard, it's really been transformational. And so I think I neglected to mention that I previously did my MD PhD training down in Philadelphia at the University of Pennsylvania. I went and did pediatrics general residency at Boston Children's, and followed by pediatric hematology oncology fellowship at the combined Dana-Farber Children's Program. But I really think that it wasn't till I arrived here and was given the opportunity to start truly pursuing my own independent problems that I wanted to answer. That was what was really transformational, because in all my prior phases of training, everything was done in the context of what someone else's overarching goals were. And while those presented tremendous environments and opportunities for learning skills, learning how to think about medicine and science at the same time, they weren't what really drove my passion. And so the ability to have this program that invests this many resources into you to let you develop, it really was transformational. And I think this has honestly just been the most exponential growth in my career in the last 20 months or so that I've been here, 18, 20 months that I've been here. And so I don't know for the other three of you what whether the experience has been similar or different and in what different ways, but I thought I'd jump in since I'm certainly the closest to our prior avenues of training.
So Ho-Jin, well, I can acknowledge that I'm the farthest from training, and that's the oldest, but that's okay. But I actually had a very similar experience and feelings around the program where I really, for me, joining as a clinical investigator here, it really was the first time that I had sort of autonomy and this level of responsibility that you just don't have when you're sort of a grad student or a postdoc. And just, it was an amazing first time that you have this opportunity to develop and probe scientific questions independently. And I really found that ability to drive the opportunities, to drive the collaborations was just incredibly energizing.
Yeah, it's really fun when the training wheels come off and you get to actually go and do it. And I think that was the fun part of becoming an independent fellow for me.
Yeah, and maybe as the person that most recently kind of transitioned out of it, I would add this, that the training wheels are nice, believe me. Mike can tell you, he had me under his wing for a long time and I was trying to break out and he was like, no, stay in. And he was right. So listen to Mike. Mike knows best. So the thing is that the clinical investigator position, when you've made that jump, when you really are on the hook and when, it's time to show, it's go home or show up or go home. I don't know. But that position, that time where a lot of the pressure is actually off because you're given this unique opportunity where you have a lab, but without all of the pressure of having to make that lab work right now. And you have this position where you can collaborate and go around and ask Phil Sharp a question, and he can tell you something that'll change your life. That is a really unique experience. And it's something that really helped me make that next step much, much, much more successful. So I can comment on that little transition there that's coming for you guys. But it makes a big difference and it's really, really important. And I think it makes a much higher quality and successful physician scientist because it's a hard thing to do. And it gives you that space to develop that technique to have a foot in both worlds.
The four of you are so unique in really doing hardcore, amazing science and also seeing patients. And so one of the attendees is asking what I think is a terrific question, and that's, how do you talk about your research to your patients, or do you talk about it to your patients?
I can answer that one because we had recently an experience. So some of the grant funding agencies actually ask you to have patients involved in the grant and involved in the proposal. So I had that opportunity where I'd actually would go and talk to patients about what we're doing in the lab. And it's very different. You have to really think about how to try and make things understandable. And it's a challenge, but I really enjoyed it because trying to help somebody understand, okay, this is what we really want to do. Doesn't it sound cool? And if you see somebody catch on and get the spark, oh, that does sound like a good idea. That's a really important thing to do. And I think it's a good skill to have to be able to describe what you're doing in terms that anyone can understand, because I think it makes it better than when you're describing it for other scientists. It helps me think about how I can frame the grant in a better way, even for the scientists, to have it understandable to a broader audience.
Yeah, I completely agree, Scott. I actually have I've always talked about research with my patients, and I've been really lucky that my research ties so directly to the patients. I had mentioned the repeat biopsy program that we developed at Mass General, and there, in order for a patient to participate, We have to review sort of the rationale for this biopsy, and it's a whole consent form. And a lot of the conversation is not simply around the logistics of this, but really around why are we doing the biopsy? What are we hoping to learn? And I have to say, over the years, what I saw is that patients were becoming increasingly knowledgeable about their own diseases and wanted to know more. And so really appreciated understanding what we were going to do with those clinical specimens from them and how that research could ultimately translate to actually new therapies. Also doing clinical research, meaning clinical trials and testing new drugs. Of course, there we often, always talked about research. Again, why are we doing this? Why are we treating you with this investigational therapy? What are we hoping to find? And what is the potential impact for other patients? I think it's a really important skill, as Scott said, and patients really need to know and deserve to understand it.
Yeah, I think the fact that we are able to do such cutting edge research in the setting of being here at the Koch Institute, that has actually been a tremendous benefit to the way in which I interact with patients, because, for the types of leukemias that I see on a regular basis, modern medicine doesn't have a lot of good options. And as much as we try and as hard as we work when we're on service, there's actually a large portion of patients that we will not be able to cure. And we have to, you have to explain that to these patients and help them understand. And at the very least, even if we can't cure them, we can at least help them understand what's going on, so they don't have this fear or dread of the unknown. They at least can accept, okay, this is the process that's going on. And if we weren't involved in this type of research, and if the conversation was to just stop there, that leaves something extremely unsatisfying within you as a physician. But we can at least tell people, you know what, we are going to be able to learn from your experience, and we will do everything to make sure that you are as comfortable as possible, and that you can do as many things as you want to do for as long as you can. But ultimately, the goal is to learn from how our experience went with your particular type of cancer, and then be able to find out how the next, maybe not the next patient, but the next month or the next year or the next decade later, patients like you who walk in will be able to say, because of this patient from before, we learned this key aspect, and that is what has broken the door open for the future successes. And so I think a lot of my patients, they take a tremendous amount of pride in that, to know that, I fought my hardest against this cancer. I might not win, but the fact that I was working with people, or I was being treated by people who are working so hard on the next generation of treatment, I at least contributed to making sure the world is a better place in the future, even if I don't survive this diagnosis.
One of the things that's really unique about having the four of you having either gone through or are currently involved in the KI at MIT is we have so many undergraduates and graduate students and postdocs who don't get the experience necessarily to be able to interact with patients, and yet they can with you. And I'm wondering how trainees fit into the picture. We, as you, I'm sure, are aware, we have, in my opinion, some of the brightest, most incredible students and postdocs at MIT. And I'm wondering how they have fit into the picture, and what was it like running your own lab and mentoring some of these people?
What's, as you say, Mike, it's really amazing. I've in particular really enjoyed interacting with the MIT undergrad students. They are some of the most phenomenally bright people, and when you meet them, you can't help but think, this person might be the one who's going to found the next Facebook or the next Google, and think, well, why can't that be in the health space? Where are the transformative ideas of tomorrow going to come from? Well, they're going to come from students like we have at MIT, and so it's really sort of a pleasure and a duty to try and get them excited about problems in the clinic, and to think about some of the skills that they pick up at MIT and applying them to problems in medicine.
I was really fortunate because in the JAX lab, I had a number of UROPS research assistant, also research assistant, as well as a KI clinical investigator. And as you said, Mike, I mean, we're so lucky at MIT because, gosh, everyone I worked with, I mean, so incredibly bright and curious and motivated, and also just wonderful to work with, so fun to mentor. And, you know, many of them actually were exploring different career opportunities. Of course, were interested in medicine. Some of them actually came with me to clinic. And so they did get some exposure to what it's like to care for patients and be performing research at the same time. And I think I hope that was helpful for them.
Yeah, that's one of the most rewarding parts, I think, personally, for this type of career. When you train in medicine, you know, there's in the academic institutes, there's always kind of a hierarchy. There's a senior physician and then some residents and then some junior. So there's always kind of that training that goes back and forth. And one of the great things about the clinical investigator opportunity is you get that first chance to be kind of the person in the lab. And you have people come through to train in your lab. And so you're kind of passing on the thing that goes on in the hospital a lot, that knowledge pass. You're doing it in the lab. And I got into it. So that's one of the best parts about that, because then I've seen people go on and they've gone on to like medical school. So there's a pretty good run we've had getting people into medical school and going off. And now some of those people are out in residency. And I think maybe the first or second one may be actually out in practice now. I have to check on Scott Clark, see, because he was a resident the last time I looked, but he might be out now. So that's really cool. When you see somebody, you know, that you had a little help helping them along in their pathway, that's cool. And Mike can tell you all about that because he's a master helper in the hospital and in the lab.
So with that, I want to, there's a question from an anonymous attendee that's directly relevant here. And this person wants to know, as someone applying to MD PhD programs, how were you able to select a research interest and then a medical specialty and marry those two into your research? So Scott, you mentioned you had an interest early on in neuroscience. So I'm really curious, did everyone else have a similar type of thing? Alice, were you interested in the lung early on? And Ho-Jun, I gather you were probably pretty interested in blood cell development. I'm curious, how early on in your career did you decide, and how did you eventually figure out how to marry your research with your clinical interest?
So I think for both Alice and I, and forgive me if I'm sort of speaking for you, Alice, in the medical and pediatric oncology fields, basic science research has been so ingrained within those cultures that it's actually pretty easy and actually fairly expected that if you are going to train in either adult or pediatric oncology, you are going to undertake some sort of scientific investigation, whether it is at the clinical research level or the basic bench biology research level. And so certainly entering one of those two subspecialties really lends itself to doing this type of work. I will actually say that I always was interested in science growing up, and certainly just through various random high school internships, I was actually doing molecular cloning in high school, albeit unsuccessfully. But really, I think when I really figured out what I wanted to work on in terms of normal blood cells and malignant cancerous blood cells, really was when I was in my PhD. I was not doing any blood cell work, but I was doing gene therapy and genome editing work in the liver to try and cure hemophilia. And the biggest thing that stood out to me is that even if the liver is diseased, it's very difficult to sort of reset the system. Doing a liver transplantation is extremely difficult, and you're on immunosuppression for the rest of your life to prevent rejection. But really, the blood system was something where you could just almost instantaneously reset everything. And I think that's what really drove me to hematology oncology. So hematology being the study of blood in combination with oncology. So it wasn't until my last year of my PhD that really crystallized for me and set me on my path through the remainder of medical school. But I don't know what everybody else's experiences have been.
Yeah, I think this is something you kind of figure out as you go along the way. I mean, for me, scientific interest is sort of like a river. It kind of meanders and goes goes where it will, where it'll take you. I mean, I think when I was an MD PhD student for a long time, I didn't know what I wanted to do residency in. And as I got towards the end, I thought pathology, became clear to me would be a field where you could really think about investigation and really try and get at the basis of disease. And it wasn't until my clinical training in pathology and residency and fellowship that my research interests sort of in cancer biology and relationships between cancer and development really started to crystallize. I did my PhD in immunology and studying the immune system. So it was really a kind of a shift for me, but one that I've enjoyed.
I mean, I have wondered about how everyone does this because, we often think about traditionally MD PhDs and how sometimes, they may be working in the lab and then leave the lab for 2 weeks to do time in the hospital, there's on service time, and it's unrelated to the research. And it can sound like, gosh, that's not exactly what an MD PhD was trained to do. But more and more, of course, we have so many wonderful examples, role models here of folks who have been able to really integrate science and medicine. And exactly how you do it, I think it is a little bit different for each person. And I think for me personally, it did kind of organically evolve. I had gotten a PhD in genetics. I just loved trying to understand the genetic basis of disease. And as I mentioned in sort of my recap of my career, it just happened that, understanding the genetic basis of lung cancer was just starting to develop at the time I was also launching my career. So there was certainly a lot of, I think, fortuitous timing, but I do think that I often tell my trainees, mentees that you've got to pursue what you love to do, and hopefully there will be an intersection between what you love to do in the lab and also what you love to do in the clinic.
Yeah, I can comment. Like Mike mentioned, my PhD work was in neuroscience. And so I kind of was interested in the brain. I wanted to do a brain thing. I ended up doing cancer sort of through serendipity. And sometimes that's what happens too, where you're just going in one direction and something happens and it just changes where you're going. So the project that I did as a PhD involved, synaptic transmission and all this stuff about how neurons work. But it turned out that there was a connection to breast cancer, that patients that have a very rare form of neurological problem often also have breast cancer. So I kind of got into, oh, that's kind of interesting. What's the connection there? And then, I tried to think about like things about the brain, neurosurgery. I'm a terrible surgeon. Thank goodness, Mike, you never saw me in a surgical rotation. It would frighten even you. So surgery was right out. So that was forget it. And then I did the neurology clinical parts, and no offense to any neurologists out there, it's a very, very important field and it's very hard, but I knew that I would not be a neurologist. And so then somebody told me, hey, there's a thing called the Gamma Knife. You should go check it out. I think they do something with brain tumors. And so I wandered down to the Gamma Knife where I was, training as a medical student. And then it kind of clicked for me. And I said, oh, well, there's brain tumors. That's cancer and brain, and you can treat them with radiation. And then I was in. And so that's kind of how it all happened. Serendipity pushed me in that direction. And it was the right direction because, I really enjoy what I do and we'd like to make it work better. So that's why we're in the lab still until we get it solved.
So I will say as certainly the most senior of the people, physician scientists, what I've found interesting is how things come back around. So my PhD work was actually all in antimicrotubule drugs for cancer. And then I transitioned and became a surgeon and got very excited about tissue injury and trauma surgery. And the research we were doing basically took us back into the areas Scott alluded to of DNA damage and cancer. And I have to say, it's something we've become more and more interested in, how tissue injury in the innate immune system interacts with DNA damage in tumors to control what happens. And the strangest thing has happened over the last 6 months, and that is my clinical practice as an emergency general surgeon has almost turned into nothing but surgical oncology. So I think it's a very funny way that your clinical practice and your research interact. I really am not sure how that happened, but that's certainly what's happened recently with me. One thing that we've been talking a lot about is physician scientist is the bridge between academia and the clinic, but there's another player here as well. And I particularly want to focus this on Alice, and that's industry. And I'm really interested, Alice, if you would say a word or two about how working in the lab prepared you, or maybe didn't prepare you, to lead research at a major pharmaceutical company. Were there lessons that have been the same? Were there lessons that were different?
Oh gosh. So yeah, I mean, I think I mentioned when I spoke again about where I've been, certainly I have felt that of all the places I've been, industry really does translation well. And having thought about that, I do think that my training, basic science and clinical training, it really has enabled me to think broadly and actually think about the research with the patient front and center. It's not simply about just, elucidating mechanisms and processes related to a specific protein or pathway. It's really about how does that protein or pathway contribute to the pathogenesis of disease? And from my standpoint now in industry, how can understanding that basic pathway inform an intervention that impacts patients, and whether or not it's a drug that we're hoping to develop, or even a diagnostic test to identify patients. So I would say that all of the basic research training, but also clinical training as well, has been really critical for me to be in my current role. And again, as I said earlier, I think starting out at the KI was foundational for me.
There are two more questions I'm going to ask, and then I'm going to stop because we're going to run out of time. So I'm going to ask everyone to just limit themselves to perhaps one sentence on this, and that is, what do you think has been the biggest challenge between balancing your clinical practice and your lab work? So I would say for me, it's been time, right? Both are, yeah, neither one takes well to your devoting time to the other. But I'm curious if there have been other things that people have found other than the time pressures.
Mike, that was mine. Mine was time management.
I find there is just a little bit of a trick of getting myself in the right headspace when I go from the lab back to the the clinic or the pathology office, to get back into the diagnostic mindset versus the investigative mindset. But certainly that's something that gets a little better over time.
I mean, I think time and not being able to be in two places at once is really the major one. But as a corollary to the mentorship part, I think when you're away, but you still want to make sure the people in your lab are getting what they need from you. And so if anybody in my labs out there, I again apologize for our 1 a.m., 2 a.m. mentorship meetings when I'm on service, which I know are probably not ideal for your schedules.
Yeah, I think time is super important. If we just figured out how to clone ourselves, then it wouldn't be a big deal. But, just running back and forth here at Duke, it makes it nice because I'm not running across the river. That I was talking to the other guys about that. That made it hard. You had to really structure things. Here you can be, it's good and it's bad because you can be a little bit less stressed. It's so easy, but my clinic is right there. So that's really nice. But it is nice to go back and forth. It is just hard to have enough time to do all the things you want to do in both realms.
Lisa Schwartz, who was instrumental for many years at the KI in helping to coordinate philanthropic support for us, specifically wanted to ask Scott and Alice what leadership lessons you learned from the KI. And I'm sure I was a good negative role model for Scott by teaching him all the things not to do. What did you see? Was there a few key leadership points that you saw from Tyler or others that you think really were instrumental in your transitioning now to the more senior roles that you have?
Yeah, I guess I would maybe pick three things just in the interest of time. I think the first would be around thinking big and being innovative. I saw just a ton of that. I see that at the KI all the time. And I was struck by that even as just starting as a postdoc. The second thing would be around collaboration. And again, as sort of as a leader, I mean, collaboration is key in every field, but I would say especially in science and in drug development. And the last thing I guess I would highlight is active listening, you know, as a leader, to really be able to listen to your colleagues and use all of that information around you. So those are my three that I would highlight.
Yeah, I couldn't echo that more. I'd already put collaboration in the chat, so you stole that. That's a great, and the KI and the investigator program in particular are a model for that. Something that coming here at Duke, it was a little more side. We tried to build that, you know, more collaboration, and I think we're being successful. But you know, the experience that I had at the KI and in that position in particular really exemplify what you can do with how important it is, how it can move you forward, and you know what it can do. You know, that an environment, you know, it's such a great environment to have all of the resources right there. You know, it's this nidus of so many amazing things. You know, that's always a model that you're trying to replicate other places too, to get to that level of productivity and just ingenuity that just happens naturally when you put all those great people together.
And then the very last question that I'm going to ask each of you to just limit yourself to 1 or 2 comments on is thinking about this program, this clinical investigator program, as a catalyst for the translational components of the research that we do at the Koch. What do you think future investigators need to be able to further maximize that impact? If you had a wish list, what are the 1 or 2 things that you would tell someone, a future clinical investigator, this is what you have to take with you from your experience at the KI? You know what? Let's do this in reverse order. So let's go from youngest to oldest. Ho-Jin first.
All right. So what do I want the person who follows me to know? I can't help but think about what Alice just said a minute or so ago about really thinking big. And so I think what everybody needs to take is that with this program, you have so much more in terms of scientific resources than any other postdoctoral trainee out there. And what you really need to do if you're going to use this as a launch pad to truly make discoveries that make a difference for cancer medicine, you need to think about what you have seen in your clinical training, what is a truly unsolvable problem with the current state of medicine, and throw your resources at trying to accomplish that. And that's what you need to learn, how to think that big. I think you really hit the nail on the head, Alice, with that.
Salil, what would be on your wish list?
Take the biggest challenge you see in your clinical practice and think, how would an engineer solve that problem? Then that's the right project for the Koch Institute. That's
great. Scott, what do you think?
That's good. I think going forward, I think the advice I would give to the next generation of clinical investigators would be really seek out and foster those relationships and soak up as much as you can from the people that are around you, because it's a phenomenal group of individuals. So really take advantage of that, you know, take copious notes and really try and carry that forward, because those are the little pearls, bits of advice, you know, models for operation. You know, how does Tyler Jacks run his lab? You know, that's what you want to shoot for. What's important to Tyler? How does he think? You know, if you just take a moment and observe, because it's going on around you. If you're in the Koch Institute, it's happening right there. And just stepping back and observing how these amazing scientists operate, how they think, it's really going to help you.
Alice, bring us home.
Yeah, no, I had similar thoughts around just how amazing an opportunity is to be at the Koch and to establish these collaborations and partnerships, which that will be fundamental for your whole career. But also the last thing I guess I would say is even when you do, you know, leave the KI program, you know, I think finding those mentors, you know, right when you are here as a clinical investigator, and then even when you leave is really important. I still have mentors who I really count on, including Tyler Jacks, in fact. So that mentorship, and you will also serve that role for your trainees as well. And so those are critical relationships to focus on.
So we're already about 7 minutes over the hour. There was one question we didn't get to answer. And the question had to do with, do you like being in the hospital or better than the clinic, or the hospital or clinic better than the lab, or the lab better than the clinic? I think I can probably answer for all of us and say, I think that's the dilemma we face all the time. And the easy answer, which I think most people would agree with, is when things are going badly in the lab, I'd rather be in the hospital. And when the patients are torturing you in the hospital, you'd rather be in the lab. So I think it's really that fine balance. I think we love both. I want to personally thank all of the people who took time out of their day to attend this. We're very, very grateful to you. As you know, the Koch Institute, and especially the Clinical Investigator Program, is really came into existence through very generous philanthropy. We really have to thank all of you for that very, very much. And one more time, I'd like to particularly thank the panelists. I really want to thank Dr. Ho-Jin Lee, Dr. Scott Floyd, Dr. Salil Garg, and Dr. Alice Shaw. Two people deserve special mention who you haven't seen, but who made this all happen. And that's really Erica Reinfeld, who put the whole program together, and Rebecca Hoff, who helped us, and especially Matt Vanderheiden, our new leader, who is taking the Koch Institute forward into the next generation. Thank you all. It's been a pleasure seeing you, and we look forward to seeing you at the next With Insight.